Pick almost any estuary on the Atlantic coast of Europe. Wait for the tide to come up, and you can find a green shore crab doing something oddly deliberate. It climbs. Not far and not fast, but with a purpose you can read from a distance. It works its way up onto a rock or a piling or a clump of weed until it's sitting where the water moves fastest. Then it settles, tucks its legs, and starts to rock its body back and forth. Underneath, tucked into the fold of its abdomen, is a soft brown sac, and the crab begins to fan. If you've spent any time watching crabs, you already know this move. That's a female releasing larvae. She lifts, she rocks, she beats her swimmerets to push water through the egg mass. And the current takes her young out into the plankton, where they'll spend the next few weeks before coming back as something crab-shaped. It's one of the most ordinary sights in coastal biology. Millions of animals do it. It's been photographed and filmed and timed and described 1,000 times. Two problems with this particular crab. The first is that it's male. Open it up and you'll find the anatomy of a male shore crab, with the gonads shrunken to almost nothing. The abdomen, which on a male should be a narrow little flap, has widened and lengthened and grown a fringe of hairs along the edges, the exact hairs a female uses to hold a clutch in place. His claws are smaller than they should be. His body has been reshaped into something closer to a brooding female, and his behavior has followed the reshaping. The second problem is the sac. Crab eggs are orange and granular, thousands of visible spheres packed together like wet sand. This thing is smooth, brown, and single. It's the reproductive body of a barnacle, and the larvae he's fanning so carefully into the current belong to the barnacle. So the crab is performing maternal behavior, correctly, on schedule, for an animal that isn't its offspring, isn't its species, and isn't even in the same class of crustacean, and it's doing it while being unable to reproduce itself, ever again, because the same barnacle took that away years earlier. Which raises a question that turns out to be much harder to answer than it looks. When we watch that crab fan, who exactly is doing the fanning? The easy answer is the barnacle. It's a satisfying answer. It fits the shape of the story we like to tell about parasites. And it shows up in every documentary that has ever pointed a lens at this animal. The barnacle has taken the controls. The crab is a vehicle. Somebody else is driving. The trouble is that when you go looking for the steering wheel, it gets complicated fast. In some of these cases, we can name the structure the parasite uses to reach the host's nervous system. In others, we can measure a chemical change in the host's brain and show it lines up with the behavior. And in a lot of them, maybe most of them, what we actually have is an animal that's been colonized, damaged, chemically disturbed, and left behaving strangely, and the strangeness happens to benefit whatever's living inside it. Those two situations look identical from the outside. They're not the same thing at all. That gap is what this whole video is about, because the ocean is absolutely full of animals that are being operated by something else, and the degree of operation runs across an enormous range. At one end, you have a fish that's swimming a little too close to the surface. At the other, you have a crab that has been converted, structurally and behaviorally and reproductively, into a life support system with legs. And somewhere in the middle, there's a line where interference turns into direction, where a side effect turns into an instruction. Finding that line is the actual scientific problem, and it's a lot more interesting than the horror show framing suggests. So we're going to build this like a case file, a sequence of exhibits, each one reaching a little deeper into the host than the last, and each one carrying the honest question of what we can prove and what we're assuming. We'll start with behavior, because behavior is the shallowest layer. Nothing about the animal has been rebuilt. It's the same body, same organs, same everything. and all that's changed is what it decides to do. Then we'll go one level down into physiology, where the chemistry of the host is being altered. Then anatomy, where a body part gets removed and something else takes its job. Then reproduction, where an animal's entire future is deleted and the energy that would have gone into it is spent somewhere else. And then identity, which is the strangest layer, and the one where the question of who's in charge stops having a clean answer at all. Two things before we start, because they'll save a lot of confusion later. First, this is not about food. There's a whole other category of ocean partnership where one organism feeds another, tube worms with bacteria doing chemistry in their tissues, corals with algae in their cells, animals with gut microbes that digest things the animal can't. That's a real and fascinating subject, and it has almost nothing to do with what we're looking at. The qualifying condition here is narrower and weirder. Somebody else has to be operating the body, making it move, or making it stop, or making it build something, or making it perform a behavior that only makes sense from the outside. Feeding a host is not driving it. Second, a warning about the language we're all going to be tempted to use. Words like hijack, zombie, puppet, mind control. Those words do real damage to the science because they smuggle in an answer before anybody's tested it. A puppet implies a puppeteer with intentions and strings. What we mostly have is molecules, tissue damage, hormone systems getting pushed off their normal settings, and natural selection quietly favoring any parasite lineage whose disturbance happened to point the host in a useful direction. That's a colder story. It's also, I'd argue, a stranger one, because it means these effects can look designed without anybody designing them. All right, Exhibit 1. The barnacle that stopped being a barnacle. To understand what's wrong with that crab, you have to start with what a barnacle is supposed to be, and barnacles are already odd enough without any help. Charles Darwin spent 8 years on them. 8 years between the voyage and the book that made him famous, Buried in Barnacle Anatomy, dissecting them under a microscope until his family joked about it and his health suffered for it. Part of what fascinated him was that nobody could agree on what they were. For most of scientific history, they'd been filed with mollusks because they look like mollusks. Shell on the outside, soft animal inside, glued to a rock. Case closed. Then, in 1830, an army surgeon named John Von Thompson did the thing that solves almost every taxonomic argument in the sea, which is to look at the babies. And barnacle babies are not mollusks. They're nauplii, the classic one-eyed, three-limbed larval form that crustaceans produce and mollusks never do. Barnacles are crustaceans. They're relatives of crabs and shrimp and copepods. What they do is swim around as a larva, find a rock, cement themselves down by the front of their head, build a mineral house around themselves, and then spend the rest of their lives lying on their backs, kicking food into their mouths with their legs. That's the baseline. Hold that picture because now we're going to break it. In 1836, an anatomist looking at shore crabs described a strange, soft, yellow-brown lump attached to the underside of the abdomen. It had no shell, no legs, no eyes, no gut, no segments, no mouth. It looked like a small tumor with a stalk. For decades, it was a genuine puzzle because there was nothing in it to classify. Structures are how you sort animals, and this thing had almost no structures. And then, again, the babies. When those sacs released larvae, the larvae were nauplii, barnacle nauplii, followed by a cyprid stage, the settling stage, which is about as distinctively barnacle as anything in the animal kingdom. The lump was a barnacle. It's called Sacculina carcini, and what it does to get there is one of the most extreme things any animal does with its own body. Start with the female larva. She hatches, drifts, molts through her nauplius stages, and becomes a cyprid, which is a tiny torpedo with a bivalve shell and a set of antennules used for tasting surfaces and gripping them. So far, this is exactly what a normal acorn barnacle does. A normal barnacle cyprid crawls around on a rock, testing the chemistry, looking for a spot that other barnacles have already approved, and when it finds one, it glues its head down and begins metamorphosis into an adult. Sacculina cyprid is looking for something more specific. She wants a crab. And she wants a particular part of a crab, a soft, thin patch of cuticle, usually at the base of a hair on the body or inside a joint, somewhere the armor is only a few micrometers thick. Once she finds it, she attaches, and then she does something that has no equivalent in the rest of the animal world. She takes herself apart. Inside the cyprid shell, her tissues reorganize into a stage called a kentrogon, which is essentially a hypodermic needle with a small mass of cells in the barrel. The kentrogon grows a hollow stylet, drives it through the crab's cuticle, and injects the cell mass into the crab's blood. What's left behind, still stuck to the outside, is the shell, the antennules, the muscle, the eye, the entire body she spent her larval life building. She abandons it. Everything that made her recognizable as an animal gets left on the doorstep, and what goes into the crab is a slurry of undifferentiated cells with no shape at all. Those cells find a place in the crab's body and start to grow, and this is where the biology gets hard to picture, because what grows has no shape you'd recognize as an animal. It's a network. Fine branching threads called the interna, spreading through the crab from the inside, wrapping around the gut, threading through the hepatopancreas, following the major blood spaces, extending down into the legs. If you inject a dye and clear the tissue, what you see looks like a root system, or a fungal mycelium, or the vascular architecture of a plant. The parasite occupies the crab the way roots occupy soil. It's spread through the entire volume. This phase is slow, and it is invisible. From the outside, the crab looks completely normal. It forages, it fights, it hides, it does everything a shore crab does. Studies on rhizocephalans, which is the group Sacculina belongs to, put this internal phase at somewhere between 2 and 36 months depending on species, temperature, and host, which means an infected crab can spend a year or more walking around with a root system inside it and no visible sign of anything at all. Then the externa appears. A bud pushes out through the crab's abdomen and swells into that soft sac. And here's the first detail that should make you sit up. It emerges under the abdominal flap, the exact place, on the exact underside, where a female shore crab holds her eggs. That location is doing work. Think of it as an address. Now the barnacle needs a male, and the male has the same relationship with his own body that she does. Male cyprids in the water find the externa, land on it, and inject themselves into a pair of small chambers called receptacles. Then they dissolve down into little more than a testis. They live there permanently, supplying sperm for the rest of her reproductive life. 2 dwarf males, in most cases, is the standard complement. So step back and look at what this species has done. Both sexes hatch with functioning bodies. Both sexes throw those bodies away. What remains of a male Sacculina is a sperm factory the size of a dust mote living inside a female. And what remains of a female is a root system and a bag. Neither of them has a mouth, a gut, an eye, a limb, or a nervous system worth the name. As a lineage, they have gone further in abandoning the standard crustacean equipment than almost any animal on Earth, and they've done it while remaining, technically and provably, barnacles. The reason all of that matters is that it sets up the question properly. This thing has no brain. It has nothing that could be called a mind, an intention, or a plan. And yet what it does to the crab looks, from the outside, exactly like a plan. Here's the list. Consider it the charge sheet. The crab stops reproducing, permanently and completely. In a female, the ovaries fail to develop. In a male, the testes shrink and sperm production collapses, and it holds. That animal's reproductive life is over, whether it was mature already or still juvenile when the infection took, and killing the parasite afterward doesn't bring it back. The crab stops molting. Crustaceans grow by shedding the entire exoskeleton and building a bigger one, and an infected crab, once the externa is out, essentially quits, which makes obvious sense from the parasite's side, since a molt would rip the externa off and end the whole enterprise. From the crab's side, it means growth stops. Whatever size it was when the sac emerged is the size it stays. Male crabs change shape. The abdomen, narrow and tucked in a healthy male, becomes broad and long and segmented, with the fringe of marginal hairs a female uses to secure a clutch. The claws get relatively smaller. The copulatory appendages regress. A researcher who knows shore crabs can look at an infected male and see a body drifting toward the female template, and the crab's behavior changes to match. It grooms the externa. It cleans it. It keeps it free of fouling. It handles it the way a gravid female handles a clutch. When the externa is ready to release larvae, the crab climbs to an elevated position in moving water, adopts the release posture, and fans, and the barnacle's nauplii go out into the current on a wave of somebody else's parental care. Infected crabs have also been reported to be less aggressive and to change their burying behavior, and the fouling on their shells goes up, which is what you'd expect from an animal that isn't shedding its armor anymore. That's the case for the prosecution, and it's strong. 5 distinct systems: reproduction, growth, morphology, behavior, and life history, all pushed in directions that serve the parasite. If you wanted to write a definition of one organism running another, that list would be a good draft. Now the harder part: what's the mechanism, and how much of it do we actually know? The most striking evidence came out of anatomy work that's surprisingly recent. Researchers looking closely at where the rootlets go found that they don't just spread through the crab at random. Some of them are aimed. They converge on the ventral ganglionic mass, which is the crab's main nerve center, the closest thing it has to a central brain, and they invade it. And at the tips of those particular rootlets, in the older families of rhizocephalans, including Sacculina itself, there's a specialized structure. It's called a goblet-shaped organ, which is a very literal name, because that's what it looks like: a cup pressed into the surface of the nerve tissue. Work published in 2020 on Sacculina pilosella described their construction in detail. 3 layers of cells, unusual lamellar bodies stacked inside them, muscle cells in the middle layer, and in the funnel of the cup, the host's own nerve tissue is degenerating. A cup, made of secretory-looking cells with muscle to squeeze it, pressed against a nerve center that's breaking down at the point of contact. Nothing about that reads as ordinary growth. That reads as an injector. Then in 2023, a team publishing in the journal PeerJ examined a related rhizocephalan, Polyascus polygenius, which lives in the Asian shore crab. This species has lost the goblet organs, but it has a different specialization: rootlets that grow into the neuropile itself. And the team found something worth pausing on. When they stained for serotonin, the label showed up in the rootlets that had invaded nervous tissue, and not in the rest of the root system. Serotonin is one of the master neuromodulators in the crustacean nervous system. It's involved in aggression, posture, dominance, and reproductive behavior. A parasite concentrating a neurotransmitter specifically in the branches that touch the host's brain is about as suggestive as an anatomical result gets. So here's where the evidence actually stands, stated as plainly as I can manage. We have a parasite that grows dedicated structures which physically enter the host's central nervous system. We have structures whose architecture strongly implies secretion into that tissue. We have a neuroactive compound localized to exactly those structures. And we have a suite of host behaviors that consistently benefits the parasite. What we don't have is the connection. Nobody has isolated a specific molecule from Sacculina, shown that it's what crosses into the ganglion, blocked it, and watched the grooming behavior disappear. That experiment has not been done in this system or in almost any other, which means everything above is a chain of extremely good circumstantial links with a gap in the middle, where the proof should be. And there's a second problem, which I find more interesting than the first because it cuts at the whole idea of control. Take the feminization of infected males. It looks like an active intervention, and for a long time it was described that way. The parasite reaching in and converting a male into a female. But crustaceans have an unusual sex determination system. Unlike most animals, where the gonads run the show, crustacean sexual development is controlled by a separate endocrine organ, the androgenic gland, which produces a masculinizing hormone. Take that signal away, and a genetic male drifts toward the female form all on its own, because the female form is what the body builds by default when the masculinizing instruction stops arriving. Sacculina's rootlets are known to reach the androgenic gland along with everything else. So which is it? Is the parasite issuing a feminizing command, or is it simply destroying the male signal and letting the crab's own developmental defaults do the rest? Those two stories predict the same crab. From the outside, they're indistinguishable. One of them is direction, and one of them is interference. And 30 years of looking at feminized crabs will not tell you which you're seeing. The same doubt applies to the behavior, and here it's even sharper. Ask yourself why the externa emerges where it does. A shore crab already has a complete brooding program built into it. Hold something egg-shaped in the abdominal fold, and a whole cascade of care behavior switches on: grooming, aeration, cleaning, and eventually the climb and the fan. That program exists. It's ancient. It's sitting in the crab's nervous system fully assembled, waiting for the right stimulus. The barnacle may not have written a single line of that software. It may have simply evolved to appear at the correct address, in the correct shape, at the correct texture, and let the crab's existing machinery do the work. In which case, all the sophistication lives in the placement, and there's no puppetry going on at all. I lean toward thinking it's both, and I'll tell you why. If the externa were only a trigger for existing reflexes, you'd expect infected males to be bad at it, because a male shore crab's abdomen isn't built to brood, and his behavioral repertoire is different. Instead, the parasite also reshapes the male's abdomen into a competent brooding platform. A paper in Marine Biology in 2012 looked directly at whether host feminization gives the parasite an advantage, and the logic there is straightforward enough. A wider, fringed, female-type abdomen is simply better equipment for holding and ventilating a brood. That's a change to the hardware, made in advance, to make the pre-existing software run properly. It's harder to explain that as pure collateral damage. But notice how careful we have to be to say even that much. And notice that the crab, throughout, is doing nothing unusual for a crab. It grooms because there's something in the brood position. It fans because the thing in the brood position is releasing larvae. Every single behavior on the charge sheet is a normal shore crab behavior performed at a normal time in response to a normal stimulus. The only thing that's abnormal is what the stimulus is. That's the first real finding of this case file, and it's going to keep coming back. An animal can be completely under someone else's operation without doing anything it wasn't already capable of doing. The control isn't in the behavior. It's in who gets to press the button. Now let's go down a level, to a case where the parasite doesn't build a root system, doesn't castrate anything, doesn't touch the host's shape at all, and still gets the host killed on schedule. There's a salt marsh in Carpinteria, California, a dozen miles or so down the coast from Santa Barbara. It's small, A couple hundred acres of tidal channels and pickleweed wedged between a freeway and the beach, the kind of place you'd drive past without registering it. It is also one of the most thoroughly studied parasite ecosystems on the planet, largely because a biologist named Kevin Lafferty and a series of colleagues spent decades pulling it apart. The fish we care about is the California killifish, Fundulus parvipinus. It's a stubby little thing, is 2 or 3 inches long, olive on top, and it lives in the shallow channels that fill and drain twice a day. Killifish are famously tough. They handle heat, low oxygen, and salinity swings that would kill most fish, which is why they thrive in a marsh where the water can go from ocean normal to saltier than the ocean over the course of an afternoon. Watch the channels long enough, and you'll notice some of the killifish behaving oddly. Most of them do what small prey fish do, which is stay low, stay in the shadow of the bank, and move in short bursts. But scattered through the school, there are individuals that keep coming up. They break the surface. They roll onto their sides for a fraction of a second, which flashes the pale silver of the flank up toward the sky like a signal mirror. They shimmy. They jerk sideways for no visible reason. If you counted, you'd find these fish doing that kind of conspicuous behavior roughly 4 times as often as their neighbors. And from above, in bright California sun, over shallow water, a flashing silver side is about the most legible thing in the entire marsh. Now the important part, and this is where the case turns from an observation into a mechanism. Cut the head open on one of those fish and look at the brain. Sitting on the surface of it, and in the thin membranes that wrap it, are cysts, small, tough, transparent capsules, each one containing a single larval flatworm waiting. In a heavily infected fish, there can be thousands of them. The species is Euplorchus californiensis, and its life cycle is a masterpiece of logistics. It starts in a snail, specifically the California horn snail, which grazes the mudflats in enormous numbers. A trematode gets into the snail, takes over its reproductive system, and turns it into a factory that pumps out swimming larvae called cercariae. Those cercariae have a few hours to find a killifish. When one does, it attaches, burrows through the skin, and then makes its way inward, following nerves until it reaches the head. There it settles onto the brain, wraps itself in a cyst wall, and stops. And it waits. Because the flatworm cannot mature in a fish. It cannot reproduce in a fish. The only place on Earth where Euplorchus californiensis can become an adult and lay eggs is inside the gut of a bird that eats fish, herons, egrets, terns. The whole point of the fish is transport, and the transport only works if the fish is eaten by the right kind of animal at the right time, before the fish dies of something else. So you have a parasite whose entire reproductive future depends on a bird noticing one specific fish in a channel full of fish. And you have infected fish that flash silver at the sky. That's a suggestive coincidence. In 1996, Lafferty and a colleague named Kimo Morris went out and tested it properly. They built enclosures in the marsh, open to the air so birds could hunt in them freely, and stocked them with known mixtures of infected and uninfected killifish. Then they let the local herons and egrets do what herons and egrets do and counted what was left. The birds ate infected fish at somewhere between 10 and 30 times the rate of uninfected ones. Take a second with that number, because it's easy to skim past. That's not a modest bias. In that enclosure, being infected made a fish an order of magnitude more likely to end up in a beak. If you were designing a delivery system, and you had to move a package from a fish to a bird, a 10-fold to 30-fold increase in interception rate is an extraordinarily good result. And notice what the parasite didn't do to get it. The fish isn't sick in any way you can see. It swims normally between displays. It feeds. It grows. It isn't disabled. It isn't blinded. It isn't dragged to the surface by buoyancy or damage. There's no root system. Nothing has been castrated, Nothing has been rebuilt. Nothing has been removed. Every organ in that fish is the organ it was born with, doing the job it was born to do. The only thing that changed is what the fish decides to do with its afternoon. That's the shallowest layer of control, and in some ways it's the most unnerving one, because there's no visible damage to point at. If you handed a pathologist an infected killifish and an uninfected killifish with the brains left in the skulls, they'd struggle to tell you which was which without a microscope. The whole intervention lives in behavior. So what's the mechanism? Here we can get further than we could with the crab, though still not all the way. Work on brain chemistry in these fish has found real differences in monoamines, the family of signaling molecules that include serotonin and dopamine. One line of research measured serotonergic activity in the raphe, a brainstem region that's the major source of serotonin projections in a vertebrate brain, and found it reduced in infected fish. Serotonin in fish is heavily involved in the stress response and in the suppression of risky behavior. Turn it down and you'd predict, on general principles, an animal that inhibits itself less. That's broadly what the behavior looks like, a fish doing things a cautious fish wouldn't. That's a real result, and it lines up. It's also, on its own, not proof of anything. And the reason why is the theme of this entire video. A brain with thousands of foreign objects sitting on it is going to show altered neurochemistry whether or not anything is being directed. Inflammation alone changes monoamine levels. Physical pressure changes them. Chronic stress changes them. You'd expect a disturbed reading. Finding one doesn't tell you who wrote it. Until fairly recently, there was an even more basic problem with this case, which is worth explaining because it applies to almost every study you'll ever read about parasites and behavior. All the early evidence was correlational. Researchers caught wild fish, counted cysts, counted behaviors, and found that the two went together. That's a real finding. And it's also wide open to an obvious objection. Maybe the causation runs backward. Maybe fish that were already bolder, already spending more time near the surface, already swimming in the shallow, sunlit water, were more likely to run into cercariae in the first place. In that version, the arrow runs the other way. And the boldness is what brings on the flatworms. Same correlation, reversed causation, completely different conclusion. The only way out of that is to infect fish yourself under controlled conditions and see what happens. And in 2023, a team published exactly that in the Journal of Parasitology. They ran experimental infections with Euhaplorsis californiensis, and infected fish increased their conspicuous behaviors, which closes the loop that had been open since the 90s and moves this case from suggestive to demonstrated. The parasite causes the behavior. But that same study also produced a result I find quietly devastating for the tidy version of the story. They didn't only test Euhaplorchis. They also tested a small cyathocotylid, a different trematode, one that does not insist on the brain. And that one increased conspicuous behaviors too. Follow that out for a second. If a brain-insisting specialist and a non-brain parasite both make killifish behave conspicuously, then the behavior isn't necessarily a targeted instruction from a specialist. It might be what a killifish does when it's carrying a significant parasite burden of any kind. Feeling bad, metabolically stressed, short on oxygen, compensating for something. There's a real hypothesis in the literature that surfacing behavior in infected fish is partly respiratory, an animal in physiological trouble going to where the oxygen is highest. Or, alternatively, it means the same trick has evolved twice, in two unrelated flatworm lineages that share the same delivery problem, which under the standard criteria for identifying adaptation is one of the strongest signals you can get. Convergence is evidence of design pressure. Both readings fit the data. The experiment that would separate them hasn't been run. Now let's move from fish to something much smaller, because the amphipod version of this story is where the mechanism gets closest to being nailed down, and where the honest problem gets its sharpest illustration. In the coastal lagoons of southern France, there's a small amphipod called Gammarus insensibilis. Amphipods are the shrimp-looking crustaceans you find under any rock in the intertidal, flattened side to side, a quarter-inch long, scuttling for cover the instant you lift the rock. That flight response is the defining fact of their existence. An amphipod's whole survival strategy is to be under something, in the dark, near the bottom, all the time. Some of them do the opposite. You'll find them at the surface, out in the open, in bright water, in the middle of the day. If you disturb them, instead of diving for the sediment, they swim toward the light. They cling to floating vegetation and stay there. Test them properly in a tank with a light gradient, and the pattern is unambiguous. Infected animals are positively phototactic and negatively geotactic. They go up and they go toward the light, which for an amphipod is roughly the equivalent of a mouse deciding to sunbathe in a parking lot. The agent is a trematode called Microphallus papillorobustus, and its life cycle has the same shape as the killifish flatworms: snail, then amphipod, then a bird. The bird is the only place it can breed, and the cysts are in the protocerebrum, the front section of the amphipod's brain. Here's the detail that lifts this above a coincidence. Microphallus doesn't only insist in the head. Larvae of the same species establish elsewhere in the amphipod's body too, and the behavioral change tracks the cerebral ones. An animal carrying cysts in its abdomen and none in its brain behaves like a normal amphipod. Location predicts the behavior. Total parasite load doesn't. That's specificity, and specificity is expensive to explain away. A general sickness effect shouldn't care where in the body the parasites are sitting. The neurochemistry goes further. In 2003, a study in the Proceedings of the Royal Society by Sylvie Hellouet and Frederick Thomas looked at serotonergic systems in the brains of infected gammarids and found the serotonin labeling altered in specific brain regions, along with changes to the architecture of individual serotonergic neurons and tracts. That's structural change localized to one signaling system. And there's a companion result from a closely related system that's about as close to a smoking gun as this field has produced. In a different host-parasite pair, a freshwater amphipod and an acanthocephalin worm called Polymorphus, researchers found they could take an uninfected animal, inject serotonin into it, and reproduce the clinging behavior. No parasite, no infection, just the neurotransmitter, and the animal starts acting like it's carrying a worm. I should be precise about what that does and doesn't show, because it gets overstated constantly. It shows that serotonin is sufficient to produce the behavior. It does not show that the parasite is delivering serotonin, or that serotonin is what's happening in a natural infection. It identifies a pathway. It doesn't prove anyone's using it, which is exactly the shape of the problem in every case we've looked at so far. And it's time to stop working around it and look at it directly. Because there's an alternative explanation for all of this that's been sitting in the literature for years. It's supported by real evidence, and it's much less romantic than puppetry. The alternative goes by several names in the papers: the byproduct hypothesis, the side effect hypothesis, sometimes just pathology, which is the bluntest version and the easiest to state. Sick animals act strange. That is the entire idea. It doesn't need a parasite with an agenda, it doesn't need targeted chemistry, and it doesn't require anything to be aimed at anything. It just needs an animal in trouble. Before you dismiss that as obviously too weak to explain what we've seen, look at how much of the evidence it covers without straining. Start with what infection does to any animal, in any phylum, manipulated or not. There's a suite of behaviors so old and so widespread that biologists treat it as a single conserved package. It's called sickness behavior, and you know it personally: lethargy, loss of appetite, withdrawal, reduced grooming, a general retreat from the world. When you have the flu and you cannot make yourself care about anything, The thing flattening you is your own immune system, working deliberately. The virus never had to issue the order. It happens because an animal that rests and stops spending energy on foraging and socializing and display fights off infection better than one that pushes through. The signaling molecules of the immune response act directly on the brain. That's the design, working exactly as intended. Sickness behavior is a host adaptation, run on host chemistry, triggered by nothing more specific than the presence of something foreign in the body. Now follow that one step further, because this is the part that quietly undermines half the manipulation literature. Those same immune signals change the brain's neurochemistry as a matter of routine. Inflammation shifts how a brain produces and consumes serotonin. It changes dopamine signaling. It redirects the raw materials those molecules are built from. In other words, the immune response alters exactly the transmitter systems that every study we've discussed so far went looking for. So when researchers find disturbed serotonin in the brain of an infected killifish or an infected amphipod, they have found something that was always going to be there, whether or not any parasite on Earth had ever evolved a single manipulative trick. An infected brain is a chemically disturbed brain. That's the baseline condition. The disturbance by itself is evidence of an infection and nothing more. Then add the energy budget, because parasites are not free. A load of trematode cysts, a growing root system, a sack of someone else's eggs, all of it is built out of calories the host collected. An animal carrying that burden is running at a deficit, and animals running at a deficit change their behavior in completely predictable ways, with no parasite involved at all. Hungry animals take risks. That's one of the most reliable findings in behavioral ecology. A well-fed minnow stays in cover and lets food drift past. A starving one moves into open water, because starvation makes the math of caution stop working. Drain any small fish of a quarter of its energy income, and you will get an animal that forages longer, ranges wider, hides less, and tolerates more exposure, which looks, from a bird's point of view, exactly like an animal being served up. There's a respiratory version of the same logic. Tissue damage and immune activation raise an animal's oxygen demand, while gill damage or anemia can cut its oxygen supply. In water, the oxygen is richest right at the surface, where the air touches it. So an infected fish drifting upward may simply be short of breath, heading for the only place a fish can go to breathe easier, which happens to be the one place a heron can reach. Put those three things together, the sickness package, the disturbed neurochemistry, and the drained budget, and you have a machine for generating conspicuous, risky, out-of-character behavior in infected animals that involves no manipulation whatsoever. It will produce fish that surface too often. It will produce crustaceans that stop fleeing properly. It will produce hosts that end up in the mouths of predators at elevated rates. And it will do all of this as a side effect, the way a car with a failing engine produces smoke. The smoke isn't a signal. Nobody sent it. It's just what failure looks like from outside. This is the argument that was sitting in the literature while the zombie documentaries were being filmed. And the person most responsible for making the field face it is a parasitologist named Robert Paulin at the University of Otago in New Zealand. In 1995, Paulin published a critical review that amounted to telling his own colleagues to slow down. The field had fallen into a habit. Find an infected animal behaving oddly, note that the oddity could benefit the parasite, and declare manipulation. Paulin pointed out that this procedure manufactures a story and then files it as a demonstration, and he laid out what it would actually take to earn the word, a set of criteria borrowed from the general logic of how you identify any adaptation. Is the altered behavior complex, or is it the kind of simple degradation that damage produces on its own? Does it show what he called purposiveness of design, meaning a precise fit between the change and the parasite's specific transmission route, not just generic recklessness? Has it evolved independently in unrelated parasite lineages facing the same problem? Because convergence is the signature of selection? And the hard one, has anyone actually measured the fitness benefit? Shown that the parasite with the manipulating genotype gets transmitted more than the one without? Hold each of our exhibits up against that list and watch what happens. The killifish passes the fitness test better than almost any case ever studied. The enclosure experiment is the fitness test run in the wild with real predators, and the answer was a factor of 10 to 30. That result is the reason this small fish is in every textbook. But the specificity test is exactly where the 2023 result cuts, the one I called quietly devastating. A different trematode, one that never touches the brain, raised the same conspicuous behaviors. If a parasite that can't reach the control system produces the same output as a parasite camped on top of it, then the output may not require the control system at all. It may be smoke drifting off a fire that nobody lit on purpose. The amphipod passes the design test better than anything else we've seen. Cysts in the brain change behavior. Cysts in the abdomen don't. Sickness doesn't read anatomy that precisely. Something about position matters, and position mattering is hard to fake with general pathology. But the amphipod is exactly where the fitness data thins out. The behavior in the field, the actual differential rate at which birds take glowing and surfaced gammarids over hidden ones, is far less measured than the killifish case. And the chemistry, as we said, has a sufficiency result, but no delivery result. And Saculina, the deepest case we have, splits cleanly down the middle. The hardware changes past the complexity test with room to spare. A resculpted abdomen with brooding hairs is not what damage looks like. But the centerpiece behaviors are all borrowed. Every one of them a stock crab routine, and at least one of the parasite's most famous effects, the feminization, may be nothing more than the destruction of a hormone source followed by the host's own defaults filling the silence. Interference, wearing the costume of direction. So the honest scorecard across the 3 best-studied operations in the coastal ocean is this. Nobody fully passes. Every case has at least one pillar standing on circumstantial ground. If you came into this looking for a certified puppet, the certification office is closed. Here's where I want to be careful, though, because the tempting conclusion that the whole thing is overblown and it's all just sick animals is also wrong. And it's wrong in a way that's more interesting than either extreme. Ask what natural selection actually sees. A parasite lineage sits in a host. The infection, through nothing but ordinary pathology, makes the host slightly more likely to be eaten by the predator the parasite needs to reach. That is a transmission advantage, and it arrives for free. Selection does not care that it's a side effect. Selection cannot even tell that it's a side effect. Any heritable variation in the parasite that deepens the effect, a little more inflammation near the brain rather than the tail, a slightly earlier drain on the energy budget, a cyst position that biases the stagger upward instead of downward, gets rewarded with more descendants. Run that for a few million generations, and the side effect stops being a side effect. It gets sharpened, aimed, and timed. The smoke gets shaped into a signal, one selective pass at a time, without there ever being a moment where illness ended and manipulation began. Which means the dichotomy we've been agonizing over, byproduct versus adaptation, describes the two ends of a single conveyor belt. Pathology is the raw material. Manipulation is what pathology becomes when it happens to point at a predator, and selection is given enough time to refine it. And that reframe makes a prediction. If manipulation is manufactured out of sickness gradually, then the ocean shouldn't contain two clean categories, puppets and patients. It should contain a continuum, lineages frozen at every stage of the process, from raw, untargeted illness all the way to placements and timings, so precise, they're indistinguishable from intention. It also predicts that we will fight bitterly over individual cases, because a heavily refined byproduct and a purpose-built mechanism can be identical at the level of a dissection. The argument is exactly the residue this kind of evolution should leave behind, and the field's inability to settle it is a symptom of the mechanism itself. If you want to watch that argument at full volume, there's one parasite where the stakes got high enough that some of the best neuroscience labs in the world waded in. It is a cat parasite, of all things. And the reason it belongs in this video is that it refuses to stay on land. It keeps washing into the ocean, where it has been killing sea otters for decades, and where the debate about what it actually does to a brain stops being academic. The parasite is Toxoplasma gondii, and you have probably heard of it, because for 20 years, it was the poster organism for mind control in the popular press. It's a single-celled creature, a relative of the malaria parasite, and its life cycle has the same two-address structure as everything else in this video. It can infect essentially any warm-blooded animal, and it does birds, rodents, livestock, marine mammals, about a third of all humans, by most serological estimates. But it can only complete its sexual cycle, the part that produces the tough, long-lived eggs called oocysts, in the intestine of a cat. Every other host is a waiting room. The parasite forms dormant cysts in muscle and brain tissue and sits there, sometimes for the host's whole life. And the only way forward is for that host to be eaten by a felid. For a mouse or a rat, that sets up an obvious evolutionary invitation. And in 2000, a team at Oxford, including Manuel Berdoy and Joanne Webster, published the experiment that made the invitation famous. Healthy rats treat the smell of cat urine as a fire alarm. It's innate. Lab rats, whose ancestors haven't met a cat in 100 generations, still avoid it. Berdoy and Webster set up enclosures with scent-marked corners, cat odor in one, rabbit odor in another, and let infected and uninfected rats explore. The uninfected rats avoided the cat corner, as rats should. The infected rats didn't. Some of them appeared to spend more time in the cat corner than anywhere else. The paper's title called it Fatal Attraction, and the phrase stuck, because the result seemed to be the whole zombie story in miniature. A parasite that needs its host eaten by a cat, apparently deleting the host's fear of cats, and possibly converting that fear into something like interest. You can guess what happened next. The story grew. Researchers found that the parasite's genome carries genes for enzymes resembling the ones that make dopamine, and a hypothesis formed that the cysts were dosing the brain's reward circuitry directly. Studies reported testosterone changes, altered risk-taking, personality correlations in infected humans, statistical links to traffic accidents and to schizophrenia. By the mid-2010s, Toxoplasma had become the standard proof, cited in every article about the crab and the killifish, that parasites really can reach into a mammalian mind and steer it with precision. Then the neuroscientists arrived with better tools, and the precise version of the story started coming apart. In 2020, a group in Geneva led by researchers, including Madlena Boya and Ivan Rodriguez, ran the behavioral tests again with modern rigor, on mice, with careful controls and imaging of where the cysts actually were. The infected mice did lose their fear of cat odor. That part held, but they also lost their fear of fox odor, and foxes don't host the parasite's sexual cycle. They were less anxious across the board, more willing to explore open spaces, more willing to investigate novel objects, more willing to interact with a live anesthetized rat, which is not something a sane mouse does. Whatever the parasite was doing, it wasn't cat-specific. It looked like a general dismantling of caution, and critically, the severity of the behavioral change tracked the number of cysts and the amount of neuroinflammation in each individual brain. More infection, more inflammation, more recklessness. Read that result next to everything from earlier, and you can feel the two hypotheses grinding against each other. A general loss of fear that scales with inflammation is exactly what the byproduct camp predicts. A brain full of foreign objects and immune activity, disinhibited by damage, no targeting required. The manipulation camp answers, reasonably, that a general loss of fear still gets the mouse eaten by cats along with everything else, so selection may never have needed specificity. Recklessness is cheap, and it works. Both camps agree on the data. The fight is entirely over whether the word for it is strategy or symptom, and as of now, that fight is not settled. The human claims, for what it's worth, have fared worse. The personality and behavior correlations mostly shrink toward nothing in the largest and best-controlled cohorts. If the parasite is steering us, it's doing it too quietly to show up reliably. So why is a cat parasite in an ocean documentary? Because oocysts don't stay where cats leave them. A single infected cat can shed hundreds of millions of oocysts in a couple of weeks, and those oocysts are built like survival capsules. They persist in soil for months to years. They survive freezing. They survive most water treatment. And when rain falls on a watershed full of them, they wash downhill, through storm drains and creeks and rivers, into the sea. Salt water doesn't kill them either. They remain viable in seawater for months. Filter-feeding shellfish then do what filter feeders do, strain particles out of the water and concentrate them, and mussels and oysters and clams accumulate oocysts, the way they accumulate everything else the land sends down. On the California coast, that chain terminates in one of the most closely watched marine mammals in the world. Southern sea otters live their entire lives in the nearshore kelp. They eat huge quantities of invertebrates, and because they're a threatened population, wildlife pathologists in California have been performing detailed necropsies on stranded otters for decades. What those necropsies found, starting in the 1990s, is that a substantial fraction of dead otters carry toxoplasma, and that in some of them the parasite is the cause of death. A protozoal encephalitis, the animal's brain inflamed and studded with an organism whose life cycle runs through a litter box. A parasite of cats, killing a marine mammal that will never in its life be nearer to a cat than the beach. There's a detail in the otter work that belongs in this video's case file, and I'll state it with exactly the confidence it deserves. In the pathology surveys, otters with toxoplasma infection turned up disproportionately among the animals that had been bitten by sharks. That's an association in stranded carcasses, not an experiment, and it has confounds you can list yourself. A sick animal, from any cause, may drift into shark territory, or fail to evade, or die where sharks scavenge. Nobody has shown, and nobody honestly can show with this kind of data, that the parasite altered the otter's behavior the way it alters a mouse's. But sit with the possibility for a second, because it's the whole theme of this video, wearing a new body, an encephalitic brain, generally disinhibited, generally slowed, in an environment where the local predator is a white shark instead of a house cat. The parasite gains nothing from any of it. A shark is a dead end. A sea otter is a dead end. There are no cats in the kelp forest, and every oocyst that ends up in an otter has left the parasite's life cycle forever. California isn't the only coastline where this is happening. And the other well-documented case is worse because the mathematics of the host population leave no room for absorbing it. The Hawaiian monk seal is one of the rarest marine mammals on Earth, a species measured in the low thousands of individuals, every one of them tagged, tracked, and known to the biologists who work the islands. Hawaii had no cats until people brought them, and it now has enormous feral populations, and the island's watersheds are short and steep, which means rain moves whatever is on the land into the sea quickly and with very little filtering. Monk seals forage on reef fish and octopus and lobster along those same coasts. And since the early 2000s, pathologists examining dead seals have confirmed toxoplasmosis As the cause, again and again, more than a dozen confirmed deaths and counting, with the true number certainly higher, since most seals that die at sea are never recovered. Several of the dead were breeding-age females, and in a population this small, biologists can calculate what a single lost female means in descendants never born. The Federal Recovery Program for the species now lists toxoplasmosis among the leading threats to its survival. Sit with the strangeness of that. An endangered seal, in the middle of the Pacific, on a list of casualties belonging to a parasite whose reproductive life runs through the intestines of cats. The seal is as much a dead end as the otter. Nothing the parasite does in a monk seal's brain will ever bring it closer to a felid. The killing is pure exhaust, the byproduct of a terrestrial cycle idling on a continent-sized scale, drifting downhill into an ocean that never agreed to be part of the experiment. That's worth pausing on, because it separates two ideas that the zombie framing fuses together. Whatever toxoplasma does to a brain, it does it wherever it lands, in hosts it can use and hosts it can't. Because the mechanism is just chemistry, and chemistry doesn't check what species it's in. Out on land, in a rodent, the effect might be transmission strategy, honed by selection, or at least harvested by it. In the ocean, the same effect is nothing. Orphaned pathology, manipulation, if that's what it is, firing into the void, killing animals the parasite has no use for. The program doesn't need a beneficiary to run. That is about as far from a puppeteer as you can get. A set of molecular reflexes so mechanical that they execute even when the performance is for an empty theater. The otters, as it happens, are collateral damage in a second entirely separate manipulation story at the same time. And this one is fully marine. Sandy beaches along the same coast are packed with mole crabs, the little egg-shaped burrowers that surf fishermen use for bait. And the mole crabs carry larvae of spiny-headed worms, acanthocephalans of the genus Prophylocolus, whose adult stage lives in the guts of shorebirds. The worm's transmission problem is the standard one: get the crab into a bird. And there are suspicions, though far less evidence than in the systems we've covered, that infected crabs end up positioned where birds feed more often. What's documented beyond argument is what happens when the wrong predator intervenes. Young sea otters learning to forage eat mole crabs by the hundreds, and the larval worms, following a program written for a bird's intestine, punch through the otter's gut wall instead. In a shorebird, that behavior anchors the worm in place. In an otter, it causes peritonitis, and it has killed enough young otters that the pathologists list it as a recurring cause of death. 2 parasites, 2 life cycles aimed at birds and cats, and an otter population absorbing casualties from both, for no biological payoff to anyone, because manipulation machinery is indifferent to who actually shows up at the trap. I've kept us in ambiguity for a long stretch now, deliberately, because the ambiguity is real, and most tellings of this subject skip it. Every case so far has hinged on chemistry, and chemistry in a living brain is exactly where side effect and strategy blur together. So the fair next question is whether the ocean contains cases without the blur, cases where you don't have to argue about neurotransmitters at all, because the parasite's method is so bluntly physical that there's nothing to interpret. It does. And the cleanest one involves no brain, no behavior change worth the name, and a clam that simply loses the ability to do the one thing that keeps clams alive. On the tidal flats of New Zealand, there's a cockle called Ostrovenus stutchburii, a thick-shelled little clam about the size of a walnut, and its entire security system is one behavior. It digs. A cockle at the surface of the flat is a package waiting for a bird. A cockle an inch or 2 down in the sediment is close to untouchable. The digging is done with the foot, a muscular wedge the clam extends, anchors, and contracts to haul itself under. Strong foot, buried clam, no story. Walk one of those flats at low tide, though, and you'll find live cockles lying on the surface. Healthy-looking animals, sitting exposed on the mud in plain sight. And if you pick one up and give it the chance to rebury itself, it can't. It tries. The foot comes out, presses, flexes, and accomplishes nothing. Put it back on the mud, and it will still be there hours later, which on a flat patrolled by oyster catchers is not a survivable condition. Open the foot, and the explanation is right there, visible, countable. The muscle is packed with the cysts of a trematode called Curtuteria australis. Same architecture as every fluke story in this video. A snail first, then the cockle, then a bird. In this case, oyster catchers, whose long orange bills are built for exactly this kind of shellfish. The larvae leave the snail, swim to a cockle, and incest specifically in the foot. And as the infection accumulates cyst by cyst, the foot converts from muscle into a bag of parasite capsules. A lightly infected cockle digs a little slower. A heavily infected one can't dig at all. It gets washed out of the sediment by the tide, fails to get back under, and waits on the surface until a bird solves the parasite's problem. Robert Poulin's group, the same lab that wrote the rules about when you're allowed to say manipulation, has worked on this system for years, which is fitting, because it's the system where the word almost doesn't matter. There is no neurochemistry to argue about. Nothing is being signaled, dosed, or persuaded. The parasite reaches its bird by eating the host's escape mechanism, physically, from the inside, the way you disable a car by removing the tires. Ask whether that's manipulation or damage, and you realize the question has stopped meaning anything. It is damage, deployed at the exact location where damage becomes transport. If the killifish case is ambiguous, because sickness and strategy look alike, the cockle case resolves the ambiguity by collapsing it. Here, the sickness is the strategy. The system has a complication, though, and it's the kind of complication that shows selection doing real work, trade-off by trade-off. A stranded cockle doesn't only attract oyster catchers. Small fish and whelks work those flats too, and a cockle lying on the surface with its foot protruding gets the foot nipped. Fish crop the exposed tips of cockle feet, and when they do, they swallow the cysts in that tissue, and a fish is a dead end for this parasite. The worm needed a bird's gut, and it gets digested in the wrong stomach instead. So the strategy of crippling the foot carries a tax. The crippled foot itself gets grazed. And researchers noticed that cysts are not distributed randomly through the foot. Some settle in the tip, where the transmission benefit of crippling is highest and so is the cropping risk. Others lodge near the base, safer from fish, contributing less to the stranding. Different positions, different gambles. Inside one organ of one clam. That's variation and consequence doing all of the work. The same blind accounting that built everything else in this story. Visible at the scale of millimeters. There's one more layer, and it widens the picture from one clam to the whole flat. A tidal flat, where a meaningful fraction of the cockles are surfaced, is a different habitat than one where they're all buried. Hard shells lying on soft mud become real estate. Limpets and anemones and algae colonize the stranded cockles, animals that had no place to attach on bare sediment, and the community composition of the flat measurably shifts. Ecologists call organisms that restructure habitat like this ecosystem engineers, and in this marsh, the engineer is a parasite, working through the bodies of clams it has disabled. Keep that in view because it comes back at full scale before we're done. What looks like the private tragedy of individual hosts keeps turning out to be infrastructure, shaping where whole communities live and where energy in the system flows. The I version of the same trick is even more literal. There's a family of trematodes, Diplostomum, whose larvae specialize in an address most parasites can't use, the lens of a fish's eye. The lens is a strange place immunologically. To stay transparent, it's largely cut off from blood supply and immune patrol, which for a parasite makes it a gated community with no police. Larvae migrate there, accumulate, and the lens clouds. The fish develops cataracts, and you can see them without a microscope. A milky whiteness where there should be black pupil. Fish farmers know the look well, because these flukes plague hatcheries and trout farms across the northern hemisphere. Most of the detailed work on them comes from freshwater and brackish systems, salmon and trout and whitefish, rather than reef fish. So file this case as adjacent to the ocean rather than deep inside it, but the mechanism travels, and relatives of these worms are in coastal waters everywhere. A fish with clouded lenses is a fish in a permanent fog. Experiments in Finland and elsewhere have measured what that costs. Infected fish detect approaching threats later, react slower, flee shorter distances, get caught more easily, and spend more time feeding near the surface, where the light is bright enough to punch through their ruined optics. Each of those changes points the same direction, upward and outward, toward the gulls and terns that are the parasite's final host. And once again, there is nothing psychological to debate. The worm isn't in the brain. It never touches a neuron. It has simply fogged the cameras. And every downstream behavior is the fish's own nervous system doing its honest best with degraded input. The fish is blind, and blindness has a shape, and the shape happens to fit a bird's hunting pattern. Notice what these two cases do to our question about who's in charge. In the crab, we couldn't find the boundary between the parasite's instructions and the host's own programs. Here there are no instructions at all, and the host's programs are running perfectly. The cockle's nervous system is issuing exactly the right commands to a foot that can no longer execute them. The trout's brain is correctly steering a body whose senses have been sabotaged. Control of behavior, it turns out, doesn't require touching behavior. You can run an animal by deciding what its body is still capable of doing. Constrain the machine and the mind follows, because the mind was only ever piloting the machine. And that framing opens a question that the chemistry cases hid. If a parasite profits from its host being eaten, when does it profit? Because a trematode larva that has just arrived in a cockle or a tapeworm that has just settled into a fish is not ready. It needs weeks or months to grow to the stage that can survive in the final host's gut. A host that gets eaten during that window simply wastes the parasite, which means the parasite's interests flip partway through the infection. Early in the infection, every incentive says keep this host alive, cautious, hidden, safe. Late in the infection, every incentive says spend it. If manipulation is real, the flip should be visible. Hosts carrying immature parasites should be more cautious than uninfected animals, not less, and then swing to recklessness when the parasite matures. That's a hard prediction. The kind of two-directional signature that general sickness can't easily fake, because illness doesn't make animals safer. The clearest tests of it come from fresh water rather than salt, from a lab in Dijon working with amphipods and spiny-headed worms, cousins of the ones in those California mole crabs. Amphipods carrying immature worms hid more than uninfected animals, hugged cover harder, took fewer chances. The same species carrying mature infective worms did the opposite, swimming exposed, clinging high, offering themselves to ducks. Same host, same parasite, opposite behaviors, sorted purely by the parasite's stage of development. Protection first, sacrifice second, on the parasite's schedule, not the host's. A pattern like that is very hard to write off as smoke from a failing engine. Engines don't fail in one direction for a month and then fail in precisely the opposite direction on cue. Something is tracking time, and the something benefits from both phases. And though those particular experiments swim in ponds, the ocean owns the animal that displays the schedule more dramatically than any other. A small silver fish that half the field has built its career on, carrying a passenger that grows until the fish is mostly luggage. The fish is the three-spined stickleback, an animal about the length of your thumb that lives in coastal seas, estuaries, and the lakes and streams connected to them across the entire northern hemisphere. Sticklebacks are one of the workhorse species of evolutionary biology, studied to exhaustion for their armor plates and their courtship, and the parasite that concerns us is a tapeworm called Schistocephalus solidus, which may be the single most thoroughly experimented on manipulator in the world, because unlike almost every other parasite in this video, it can be raised, mated, and deployed entirely in a laboratory. Its life runs in 3 acts. The eggs hatch in water into a swimming speck that gets eaten by a copepod, one of the rice grain crustaceans that fill every plankton sample. Inside the copepod, the larva grows for a week or two. Then a stickleback eats the copepod, and the young worm punches out of the fish's gut into its body cavity, the open space around the organs, and starts to grow in earnest. And then a bird, a heron, or a gull, or a kingfisher eats the stickleback, and in the bird's gut, the worm matures, mates, and releases eggs back into the water, all within a couple of days. Copepod, fish, bird. 2 transfers, both by predation, which means 2 separate occasions where the worm's future depends on its current host getting caught. Start with the copepod, because the copepod experiments are the cleanest demonstration of the timing flip anywhere in biology. You can infect a copepod on a known date and track the worm's development almost to the hour. While the worm is immature, too young to survive in a fish, infected copepods become harder to catch. They freeze longer after a disturbance, move less, recover more slowly, exactly the profile of an animal, minimizing its chance of being noticed by a predator. The moment the worm crosses into infectivity, the same copepods swing the other way, resuming activity sooner, swimming more, exposing themselves. Protection during the vulnerable window, exposure after it. The schedule matches the parasite's interests so exactly, in both directions, that the sickness explanation has nothing to grab onto. Then researchers at the Max Planck Institute in Germany did the experiment this whole video has been implicitly building toward. They put 2 worms in one copepod, deliberately, with different infection dates, so that one worm was infective and the other was not. The 2 passengers now wanted opposite things from the same vehicle. The older worm profits if the copepod gets eaten today. The younger worm dies if it does. If the behavioral changes were just accumulating damage, 2 worms should simply mean more damage. Instead, in those experiments, the host's behavior mostly followed the infective worm. The suppression that a lone, immature worm reliably produces was overridden. Sabotaged is the word the researchers used. And the copepod behaved close to how a singly infected, ready-to-transmit host behaves. Whatever mechanism raises exposure, it won the argument against whatever mechanism lowers it. I want to be plain about what that shows, because it's subtle and it's the strongest single piece of evidence in this entire field. It shows the behavior of one small crustacean being set by a negotiation between 2 other organisms living inside it. With the outcome depending on their developmental states, you can't get that from inflammation. You can't get it from an energy deficit. 2 things with opposed interests fought over a body's level of caution, and one of them won. Whoever is in charge of that copepod, it is not the copepod. Now, the fish. When the worm arrives in a stickleback's body cavity, it weighs next to nothing. And for the first several weeks, it's a quiet tenant. The fish schools, feeds, flees, behaves like its neighbors. The worm, meanwhile, grows faster than any tissue the fish owns, absorbing nutrients across its whole surface, directly from the fluid around the organs. And it does not stop. Worms reach hundreds of times their arrival weight. A single tapeworm can come to rival a substantial fraction of the fish's own mass. And multiple infections push further. Heavily infected sticklebacks swim around visibly deformed. Bellies stretched taut and squared off around coiled worm, an animal, the approximate shape of a fish, wrapped around a payload that is not fish. The energetic arithmetic is brutal. Every calorie of that worm was foraged by the stickleback, swallowed by the stickleback, digested by the stickleback, and then diverted before the stickleback's own tissues could use it. The behavioral change arrives on schedule, and the schedule is the worm's. Laboratory work has repeatedly found that the shift tracks the worm's size, kicking in around the threshold, roughly 50 milligrams, about the weight of a grain of rice, where the worm becomes capable of surviving in a bird. Below the threshold, infected fish behave nearly normally. Above it, they transform. They feed out in open water instead of in the weeds. They rise towards the surface. Startle them with a simulated heron strike, and they dive like any fish. But they come back up sooner, resume feeding faster, drift back into the open while their uninfected schoolmates are still hiding. They tolerate approaches from predators and from nets and from researchers' hands that a normal stickleback would never allow. Fishermen and field biologists have known for a century that if you can catch a stickleback with your bare hands, it's probably carrying something. Two explanations are on the table, and by now you can recite them yourself. The worm may be releasing something into the fish, and there is real evidence of altered stress physiology in infected fish, shifts in the hormonal axis that governs threat responses, or the fish may simply be starving in plain sight, its risk calculus legitimately rewritten by an energy deficit no ordinary hunger could produce. Boldness as the correct decision of a body being robbed. The size threshold timing favors the worm having some say. The arithmetic favors the robbery mattering regardless. The two aren't exclusive, and in this system more than any other, researchers suspect the worm's cheapest trick is the theft itself. Manipulate the budget, and the behavior follows by itself. No neurochemistry required, because a hungry enough fish will always go where the food and the light are, and that place is where the birds hunt. The robbery has a second consequence, and it connects this case backward to the crab. Female sticklebacks build eggs out of surplus energy, and an infected female has none. Egg production falls, and in heavy infection stops entirely. Males fare no better. Nest building and courtship are expensive, and heavily parasitized males often simply don't breed. Nobody castrated this fish. There's no root system in its gonads, no severed hormone gland, no anatomical intervention at all. The reproductive shutdown is pure accounting. Sterility as a line item, the same end-state Saculina engineers surgically, reached here by nothing more sophisticated than theft on a sufficient scale. Two parasites, two utterly different methods, one identical outcome. A host that lives on, works on, forages on, and puts everything it earns into another species' offspring. Keep that convergence in mind, because it's the shape of a pattern this video is about to run into everywhere. For a parasite that transmits by predation or lives by extraction, the host's reproduction is the one expense that never pays, and lineage after lineage, by chemistry or surgery or simple starvation, finds its own way to cancel it. There's one more fact about Schistocephalus, half legend in the field, that says more about the nature of these systems than any behavioral assay. In the 1940s, a parasitologist named James Smith wanted to study the worm's final act without dealing with birds, and found he didn't need the bird at all. Take a mature worm out of a stickleback, put it in warm nutrient fluid at bird body temperature, around 104 degrees Fahrenheit, and it wakes up, matures, and breeds. The technique, refined since, still underpins the entire laboratory culture of this species. Worms mated in glassware, in little mesh-bottomed containers, standing in warm baths, no heron involved. Think about what that means. The destination this parasite spends months of manipulation trying to reach, the goal of the copepod's suppression and the copepod's sabotage, and the stickleback's slow-motion sacrifice, is, biologically speaking, a temperature. The entire Baroque pipeline of altered behavior exists to deliver the worm into any warm, wet, nutrient-rich space. And a bird's intestine is simply the only such space that nature reliably provides at the top of a food chain. The manipulation looks like strategy from outside. From inside, it's a thermostat. The worm is seeking 104 degrees, and it has learned, in the way that lineages learn, that the warmth is on the far side of a beak. Everything we've examined so far has left the host physically recognizable. Chemically disturbed, energetically gutted, blinded, crippled, sterilized, but structurally itself. A fish shaped like a fish, a cockle with all its parts. The next exhibit crosses a line none of the others touch. It's the case where the parasite stops operating the host's equipment and starts replacing it, where you can open an animal's mouth and find that a piece of its body is now a different animal looking back at you. In the Gulf of California, fishermen hauling in rose snapper occasionally get a surprise when they go to unhook the catch. The fish opens its mouth, and where the tongue should be, there's a face. Two small black eyes, a segmented pale body about the size of the top joint of your thumb, a set of gripping legs, looking out from the floor of the fish's mouth like something that has moved in. Because it has. The animal is Cimothoa exigua, an isopod, which puts it in the same broad group of crustaceans as the pill bugs in your garden, and its common name, the tongue-eating louse, is one of the rare cases where the common name undersells the biology. What this animal does was described formally in 1983 by two researchers, Richard Brusca and Matthew Gilligan, working with snapper from Mexican Pacific waters. And their paper documented something that had never been confirmed for any parasite before or since: the complete functional replacement of a host organ by another species. The sequence goes like this. Juvenile isopods swim free and enter a fish through the gills. They're males at this stage, all of them, small and mobile, and they settle among the gill arches. Cymothoids are protandrous hermaphrodites, meaning they start life male with the option of becoming female later, and the option gets exercised by circumstance. On a fish with no female present, one male, usually the largest, changes sex, grows, and relocates. She crawls forward out of the gill chamber and into the mouth, and clamps onto the base of the tongue with 7 pairs of hooked legs. A fish tongue is a muscular, bone-stiffened ridge on the floor of the mouth, nothing like yours, and it's a working organ. The fish uses it to hold prey against the teeth on the roof of its mouth and help move food back toward the throat. It has a rich blood supply, and the blood supply is what the isopod is there for. She pierces the tongue and feeds on the blood flowing through it. And as she grows, the demand grows, and the tongue, its circulation increasingly diverted and throttled, begins to shrink. Over time, it withers back to a stub. In any ordinary parasite story, that's where things would end. A mutilated host and a fed parasite. Here it's where the strange part starts. The isopod doesn't leave the ruin. She turns and grips the muscular stub with her rear legs, positions her body forward along the floor of the mouth, exactly where the tongue used to lie, and stays, permanently. And the fish uses her. It presses prey against her armored back the way it pressed prey against its tongue. It works food down its throat over her body. Brusca and Gilligan examined the fish they collected and found them feeding in apparently workable condition. Some studies since have measured costs, slightly lower body weights, slower growth in some hosts, but the arrangement functions. The fish operates a prosthetic tongue that is alive and eating and female, and while she sits there, she mates with the males back in the gill chamber and broods her young in a pouch on her underside, in the wet shelter of a mouth that belongs to someone else. Set this case against every other exhibit in the file, and notice how completely it inverts them. There is no behavioral change worth mentioning, no neurochemistry, no recklessness, no altered relationship with predators. The parasite has no interest in this fish being eaten ever, because for this parasite the fish is home, and home never gets spent. The snapper hunts, flees, and schools like any snapper. All the sophistication that other parasites spend on steering, this one spends on becoming furniture. And yet, if the question of this video is whether another species can run part of an animal's body, here the answer isn't metaphorical. When that snapper feeds, its brain issues motor commands to the floor of its mouth, and the thing that moves, the physical object performing the function of tongue in the loop between the fish's intention and the fish's meal, is a crustacean. The fish is running the isopod. The chain of command points the wrong way. The host's nervous system has incorporated a foreign animal into its own body plan and doesn't know it, because nothing in a fish's brain checks whether the tongue is still made of fish. That last point deserves a second, because it's the mechanical heart of this entire subject, stated more nakedly here than anywhere else. The snapper's brain doesn't have a concept of its tongue. It has reflex loops, pressure here, response there, food against palate, swallow. The isopod satisfies the loops. She's roughly the right shape, roughly the right stiffness, in exactly the right place, and that is all the system ever asks. There's no audit. No immune tribunal reviews the mouth and declares its contents self or other. The body's practical definition of self turns out to be whatever is where it should be, doing roughly what it should do. Sacculina passes as an egg mass and inherits a crab's parental care. The isopod passes as a tongue and inherits its job. Neither of them fooled a mind because minds were never consulted. They fooled an interface. Some housekeeping on this animal, because it became briefly famous online and the fame distorted it. Photographs of snapper with isopod faces in their mouths circulated for years with captions implying horror movie rarity or danger. The animal is neither rare nor dangerous to you. Cymothoa exigua is one of hundreds of species in its family. Tongue-replacing behavior has now been reported from several of them, in different fish, in different oceans. Another instance of the convergence that signals a workable strategy rather than a freak event. Related cymothoids don't bother with the tongue and simply clamp onto gills or skin as more conventional bloodsuckers, which suggests the tongue specialist came from ancestors like those, lineages that crept, generation by generation, toward the one perch inside a fish where a parasite can be shielded, fed, and extraordinarily useful. And a person who puts a finger in the wrong snapper's mouth risks, at most, a pinch. She has no interest in anything that isn't a fish's bloodstream. It's worth asking the survival question from the fish's side, because at first glance the arrangement seems too costly to persist. Why hasn't selection armed snapper against this? Part of the answer is likely that it can't afford to. A mouth and gill chamber must stay open to flowing water. The same architecture that makes a fish makes it enterable. Part is that the cost, while real, is survivable, and selection is weakest against harms that maim rather than kill. And part may be the cold arithmetic we keep meeting. A snapper, with a replaced tongue, still feeds, still grows, and unlike nearly every other host in this video, still breeds. By the standards of the animals we've been watching, the snapper got off easy. It lost an organ and kept its future. Which is precisely what the next hosts don't get to keep. Because the strategy that Sacculina applies to one crab with surgical roots, and the tapeworm applies to one stickleback through theft, exists in the ocean at industrial scale. Whole guilds of parasites whose entire relationship with their hosts is the cancellation of reproduction, and in at least one California marsh. The science has now weighed them, all of them. And the number that came back rearranged how ecologists think about who is actually in charge of an estuary. Go back to Carpentaria, the salt marsh, where the killifish flash at the sky, and this time ignore the fish. Look down at the mud. It's paved with snails. California horn snails, dark spiral shells, about 1.5 inches long, grazing the sediment in densities that can reach hundreds per square yard. one of the most abundant animals in any Pacific estuary. They are the marsh's great intermediate host, the first stop for the trematode flukes whose later stages we've already met in the killifish, and what the flukes do to the snails is the reproduction story stripped of all subtlety. A fluke egg arrives in the marsh in bird droppings. It hatches, finds a snail, bores in, and heads for the gonad. And there it does something categorically different from anything we've seen. It doesn't insist and wait, it colonizes. The parasite converts the snail's reproductive organ into a factory of its own tissue, a mass of sac-like or worm-like bodies that do nothing but bud off swimming larvae, the cercariae that will go hunting for killifish. The snail's own reproduction ends completely, usually forever. Parasitologists don't reach for softer words here. The technical term in the literature is parasitic castration, and it's exact. The snail is not sick. Its survival machinery, feeding, crawling, predator avoidance, is left carefully intact, sometimes for years, while the machinery of its future is demolished and replaced. Think about what that snail now is. It eats every day, converts algae and detritus into energy, and spends that energy maintaining a body whose biological purpose has been deleted and overwritten. On warm days, infected snails shed clouds of larvae into the tidewater, hundreds upon hundreds of them, day after day, season after season. Every one of those larvae was built from calories the snail grazed. The animal has been converted from an organism into a production line, a self-feeding, self-repairing, predator-avoiding machine for manufacturing another species, still crawling around its mudflat years after its own lineage has, in every sense that matters to evolution, already ended. Some biologists describe castrated hosts with a phrase that sounds like rhetoric and isn't: the walking dead, alive as an individual, extinct as an ancestor. The scale of it is what forces the rethink. Survey after survey in these marshes has found trematode infection running through the horn snail population at rates that climb steeply with age, because a snail can't be cured and can't outgrow it. Every year of life is another year of exposure. Among the biggest, oldest snails on a heavily birded flat, infection is not the exception. Much of the marsh's snail biomass is, functionally, fluke. The occupation even shows up in the architecture of the shells, though not in the direction you'd guess. In a number of snail systems around the world, castrated snails go on growing, and some grow larger than any uninfected snail in the population, a phenomenon parasitologists call gigantism, and its meaning has been argued about for decades in a miniature version of this video's central fight. One reading says the parasite is doing it. A bigger snail is a bigger factory, more tissue to convert, more surface to graze with, and a colony that will occupy this body for years profits from expanding the premises. Another reading says the snail's own physiology is doing it with no guidance at all. Reproduction is expensive. The parasite has canceled that expense, and the freed energy has to go somewhere, so it goes into flesh and shell by default, the way a plumbing system reroutes when one branch is capped. A third notes that the hormonal wreckage of a demolished gonad could distort growth as a pure side effect. All three explanations fit the data. The outcome varies from system to system. Some hosts grow giant, others are stunted. Sometimes the same parasite produces each in different conditions. And so even here, in the least glamorous corner of the subject, the question of who a change serves and whether anyone is steering it stays wide open. The pattern you should recognize by now: the closer you look at any of these systems, the harder it becomes to find a fact that belongs to only one owner. And it's not one fluke. Around 20 different trematode species use the horn snail in these estuaries, each with its own final destination. Different birds, different fish, different crabs, and only one parasite colony can hold a snail at a time. Which sets up something almost nobody outside the field knows exists. Competition, invasion, and defense, fought between parasites inside the body of a snail. When larvae of a second species bore into an occupied snail, the residents don't simply share. Dominant species destroy subordinate ones, physically attacking their tissue, and the occupations have a ranked order, a hierarchy of which fluke evicts, which stable enough that researchers can predict the outcomes. Then, a little over a decade ago, work led by Ryan Hechinger and colleagues found something in those snails that pushed the strangeness up a full level. In some trematode species, the colony inside a snail contains two castes. Most members are the standard form, bloated with developing larvae, barely mobile. Pure factory floor, but scattered among them are small, thin, fast-moving individuals with oversized mouth parts that never reproduce at all. Put an invading parasite in with them and the small ones attack it, latching on and tearing at its body. They're soldiers, a division of labor, defenders and reproducers, genetically identical clone mates splitting roles the way ant colonies do, garrisoned inside the gonad of a snail, defending their conquered organ against other parasites. The snail's body, in other words, is occupied territory, valuable enough to hold with a standing army, contested enough that an army is worth building. Once you've absorbed that image, the next question asks itself, if snail flesh is territory, if hosts are resources, parasites conquer, farm, and defend, then how much of the marsh, by weight, is actually parasite? For most of the history of ecology, nobody asked, because the assumption answered itself. Parasites are tiny, hidden, negligible, a rounding error riding on the real economy of plants, grazers, and predators. In 2008, Armand Curis, Kevin Lafferty, and a large team published, in the journal Nature, The Accounting. They had spent years literally weighing the parasites in 3 Pacific Coast estuaries, Carpentaria among them, dissecting thousands of animals, tallying every fluke and worm and body snatcher, and summing the mass. The parasites outweighed the birds. Outweighed, and by a wide margin. In those estuaries, the combined biomass of trematodes alone, animals with no mouths to speak of, no eyes, most of them living as colonies inside snails, exceeded the combined biomass of all the birds in the system, the herons and egrets and oyster catchers that any visitor would identify as the marsh's ruling class. The annual production of larvae pouring out of the infected snails was comparable to the production of whole, free-living animal groups. The hidden economy was one of the mainline items, a river of energy flowing from sunlight to algae to snails, and then sideways, out of the visible food web entirely, into the manufacture of parasites. A few years earlier, some of the same researchers had rebuilt the marsh's food web on paper, the full map of who eats whom, and then added the parasites in every host link and life cycle passage. The web nearly doubled in connections, and the parasites were involved in the majority of the links in the completed map. Follow what that means for the theme of this video. The energy that funds a heron's existence travels, in meaningful part, through manipulated bodies, through snails converted into factories, through killifish flagged for capture, through crippled cockles on other flats, delivered to the birds by parasites that need the birds. The manipulation isn't a sideshow curiosity in this system. It is load-bearing infrastructure. The marsh, as an energy economy, partly runs on it. If all of this still feels like the private weirdness of one California wetland, it isn't. And the proof is on fishing boats. Rhizocephalans, Sacculina's family, the root barnacles that opened this file, infect commercially important crabs across the world's oceans, and everywhere they do, they bring the same policy: total, permanent castration of the host, redirection of its energy into parasite larvae, and a crab that stops molting and stops growing. In the fjords of Alaska and British Columbia, a rhizocephalan called Bryorosacus infects king crabs, the same animals that fill seafood cases, and in some enclosed fjord populations, surveys have found infection rates high enough to matter to the fishery. Entire local stocks, in which a large share of crabs are sterile vehicles for barnacle larvae, worthless to the fishery and finished as breeders. In the Gulf of Mexico, a related species, Loxotholacus texanus, does the same to blue crabs, and infected animals are recognizable at a glance, undersized, misshapen, carrying the telltale sac where a female's eggs would ride. And in Chesapeake Bay, the story acquires a distinctly modern chapter. The Chesapeake's mud crabs historically had no rhizocephalan. Then, in the 1960s, oysters were transplanted up from the Gulf of Mexico to restock Chesapeake beds. And riding with the oysters, in crevices and shell interiors, came Gulf mud crabs, and inside some of those crabs came Loxotholacus panopei. It established, spread, and today infects Chesapeake mud crabs at rates that in some creeks run high, a parasitic castrator operating on a population with no co-evolved defenses, delivered by barge. We tend to file invasive species as animals that escape and outcompete. This one is stranger, an invasion of reproductive sabotage, a body-snatching lineage that humans shipped up a coastline without ever knowing it was aboard, now quietly deleting the futures of crabs whose ancestors never met it. Stand back from all of it, the snail factories, the soldier casts, the biomass ledger, the sterilized fisheries, and the picture that emerges is not the one the word parasite usually paints, of small thieves skimming from the edges of the real world. In these systems, the hosts are the resource base and the parasites are a governing layer, extracting, redirecting, defending territory, and shaping where the energy of entire ecosystems goes, mostly invisibly, through the mechanism this whole video has been circling, the takeover of budgets and bodies. So it's time to stop circling. We've reached the last layer, the one I promised at the start, where the question of who's in charge stops having a clean answer, and it stops because the question itself starts to break. In 1982, Richard Dawkins published a book called The Extended Phenotype. And buried in its argument is the idea that dissolves our question. A phenotype, in the standard telling, is the physical expression of genes, your body. Dawkins pointed out that nothing in the logic of evolution confines a gene's effects to the body it sits in. A beaver's genes build the beaver, and then, through the beaver's behavior, they also build the dam and the lake behind it. The lake is as much an expression of beaver genes as the teeth are. Genes reach out through behavior into the world, and selection judges them on everything they touch. Now apply that to a parasite whose world is another animal. If a beaver's genes can express themselves as a lake, then a fluke's genes can express themselves as a fish's swimming pattern. A barnacle's genes can express themselves as a crab's widened abdomen and its climb into the current. The host behavior becomes, in the precise technical sense, part of the parasite's phenotype, a trait of the barnacle that happens to be made of crab. When selection favors barnacle lineages whose crabs climb higher and fan better, it is sculpting crab behavior exactly as directly as it sculpts barnacle rootlets, through the same mechanism, heritable variation, and differential success, merely routed through a second body. That's the frame that finally lets us answer the question we started with on that estuary rock. Who is doing the fanning? Ask it as a question about anatomy, and the answer is the crab. Its muscles, its nerves, its ancient brooding reflexes, nothing in the motion that isn't crab. Ask it as a question about information, about which genome the behavior exists to serve, and which genome's variation shapes its details across generations. And the answer is the barnacle. Both answers are completely true. They're answers to different questions that our language, built by and for organisms that assume they own themselves, insists on treating as one. The fanning is crab hardware running barnacle fitness. The sentence sounds paradoxical only because we've been assuming that a body and its beneficiary must be the same organism. In the ocean, over and over, they aren't. Look at how deep the nesting actually goes in that one animal on the rock, because it's worth spelling out slowly. Innermost, there are two dwarf males, reduced to almost nothing but sperm, living inside receptacles in the body of the female barnacle. The female barnacle is a root system and a sac, living inside and upon the crab. The crab, its chemistry adjusted, its shape resculpted, its behavior serving the sac, carries all of them up the rock. One climbing body, 4 genomes aboard, and the genome steering, to whatever degree steering is happening at all, belongs to an animal with no brain, while the genome that owns the brain contributes the movements but not the purpose. Ask which of these organisms is the individual here. Draw the boundary of the animal on the rock, and every line you try cuts through something. The crab alone? Then the thing that determines the crab's behavior lies outside the animal. The crab plus the barnacle? Then the individual is 2 phyla at once, born in different years, and only one of them can reproduce. Biologists have a genuine, unresolved literature on this, and not because of parasites alone. The clean concept of the individual, one genome, one body, one set of interests, phrase at every edge you inspect. Nearly every animal, you and the crab included, runs partly on a microbial workforce it didn't inherit in its genome. Colonial sea creatures blur where one individual ends and the next begins. What the manipulators add to this picture, and it's the sharpest edge of all, is conflict. The microbes in a gut mostly profit when their host profits. Saccharina profits when the crab's own evolutionary interests are erased and its body redirected wholesale. An infected crab is something with no good everyday word for it, short of a partnership and well past a chimera. A body whose parts answer to rival owners and whose observable behavior is the settlement, imposed rather than negotiated, of that rivalry. Here's the reframe that settles out of all this, and I think it's the single most useful idea in this entire subject. We've been asking case after case whether the parasite is really in control, as if control were a substance, as if somewhere in the crab or the killifish there were a steering wheel that either the host holds or the parasite grabs. There is no wheel. There was never a wheel. What an animal's body actually is underneath the impression of unified purpose is a federation of mechanisms, reflex arcs, hormone axes, motor programs, feeding drives, brooding routines, each one triggerable by particular inputs, none of them equipped to verify where its inputs come from. In an uninfected animal, all the triggers happen to be pulled by the animal's own physiology, and the federation moves with such coherence that we name it a self, and assume the coherence is enforced from a center. It isn't. The coherence is just the default pattern of inputs. It has no guard. That is the discovery sitting under every exhibit in this file, the thing the parasites collectively prove about their hosts, and honestly about all animal bodies, ours not exempted. The brooding program of a crab will run for anything egg-shaped at the right address. The swallowing reflex of a snapper will operate anything tongue-shaped at the right position. The risk calculus of a stickleback will faithfully recompute for whatever energy budget it's handed, with no line of code to ask why the budget collapsed. Serotonin will do serotonin's job in an amphipod's brain regardless of which organism's metabolism produced it, because molecules carry no signatures and receptors check none. Every port in the machine accepts input from anyone who speaks the protocol. Bodies, it turns out, were never fortresses with a commander inside. They're open architectures secured by nothing but the historical improbability of anyone else learning the passwords. Parasites are what happens when something has unlimited evolutionary time to learn the passwords. Seen from this angle, the manipulators stop looking like invaders seizing power and start looking like something closer to water finding cracks. No individual parasite knows anything. Saculina's larva follows chemical gradients into a crevice its ancestors' descendants survived entering, and there is no scheme anywhere in it. The fluke in the killifish brain composes nothing. Its lineage got filtered, Cercaria by Cercaria, over spans of time that make human history a rounding error, until the only variants left were the ones whose effects on fish brains happened to end in a heron. The learning is real, but it's lineage learning, blind, cumulative, stored nowhere except in what survived. When we watch the crab climb and feel the presence of intention, we're right that there's design in the scene. The only question is where it lives. It doesn't live in the barnacle. It lives in the filter, in millions of drowned failures. And the barnacle on the crab is simply the shape of what passed through, which leaves one last piece of business before we close the file. If the deep claim is right, if there's no wheel and control is just access to triggers, then the century of argument between puppetry and pathology should resolve case by case into something more precise than either. It does. And walking through that resolution, what we can now actually say about each animal in this file, and how sure we're entitled to be, is the right way to end, because the resolution is stranger than the headline it replaces. Start with what proof would even look like, because the standard matters and it's higher than almost anything in this video has met. To demonstrate manipulation the way a pharmacologist would demand, you need the full chain. Identify a specific molecule the parasite produces. Show it reaches the host tissue that governs the behavior. Deliver that molecule alone, no parasite, and produce the behavior. Block it in a real infection and watch the behavior vanish while the parasite sits there intact. 4 links. Every case in the ocean is missing at least 2 of them. The closest any animal system has come is not marine, and it's worth a short detour to see what the finish line looks like. Horsehair worms are long, wiry parasites of insects. Their larvae grow inside crickets and grasshoppers, and the adult worm must breed in water, which is a problem, because crickets avoid water. Infected crickets don't. At the worm's maturity, and at night the insects walk to ponds and streams and jump in, whereupon the worm exits its drowning host and swims off to mate. In the 2000s, a French team led by David Biron and Frédéric Thomas did something no one had managed in any manipulation system. They ran molecular surveys on the heads of crickets during the manipulation itself and found the worm producing proteins from families the insect's own nervous system uses for its development and signaling, molecules shaped like the host's own vocabulary, present precisely while the behavior ran. Molecular mimicry, the parasite speaking the host's chemical language, caught partially in the act. It's still not the full 4-link chain. Nobody has synthesized the mimic, injected it, and marched a healthy cricket into a pond. But it's a hand on the doorknob, and it tells us the endgame of this kind of evolution is exactly what the identity argument predicted. Forging the machine's passwords, one protein family at a time, with nothing ever needing to be overpowered. And the ocean? The ocean has a horsehair worm of its own, a genus called Nectonemae, whose larvae grow inside crabs and shrimp, hollowing out the same niche its freshwater cousins fill in crickets. Whether it moves its hosts, whether an infected crab does anything a healthy crab wouldn't, essentially nobody knows. The animal is that little studied. Specimens turn up by accident in trawl catches and lab tanks, and the basic natural history remains unwritten. I bring it up partly for honesty because it marks the true boundary of knowledge here, and partly because it's this subject in miniature. The best proven mechanism in the field lives just on shore. Its marine sibling swims in near total scientific darkness, and the gap between them is a gap in looking. The marine cases, meanwhile, are being pushed down toward molecules from the anatomy side, the goblet organs pressed into crab ganglia, the serotonin localized in nerve-invading rootlets. Those are the outward visible ends of chemistry nobody has yet caught in transit. Genomes are coming, rhizocephalins and their hosts are being sequenced, and somewhere in that data, if the manipulation is real, are genes for secreted molecules with no housekeeping excuse to exist. Molecules shaped to fit crustacean receptors the barnacle itself no longer possesses. Finding one, purifying it, and running the four-link chain in a tank of shore crabs is a brutal, unglamorous project that would take years, which is exactly why it hasn't happened, and it is the single experiment that would move this entire subject from circumstantial to closed. Someone will do it. The tools finally exist. The Sacculina question, open since anatomists mistook the parasite for a tumor in the 1830s, is likely to be answered within the working lives of scientists already in the field. While we wait, I want to put the judgment call to you. Because through this whole file, the hard part has never been the facts. It's been where to draw the line. And line drawing is a thing audiences here are unusually good at. So here's the question, and I mean it as a real one, not a rhetorical flourish. Take the cases you've now seen. The barnacle that reshapes a crab and inherits its parenting. The fluke that demonstrably gets its fish eaten but maybe only by making it feel bad. The cysts that turn caution off in an amphipod's brain. The worm that runs a copepod on a timetable and outvotes its own rivals inside the host. The cockle parasite that just eats the escape organ. The isopod that becomes the organ. Where in that lineup does interference end and direction begin? And what single experiment would move your line? Put it in the comments, name the case, and the test. I go through them and I'll say plainly that on questions like this one, where the fight is about standards of evidence rather than data, the arguments that show up in comment sections are routinely sharper than the ones in review articles. Because you're not defending anybody's grant. If you've worked in a lab or on a boat, or you've pulled a crab off a line and found the sack under its tail, I especially want your answer. And if this kind of story is why you're here, the slow kind, where a question gets taken apart honestly instead of shouted, subscribing is how you keep it coming. And a like on the way out is a small thing that helps this channel keep making them. Now let's close the file properly, exhibit by exhibit, saying exactly what the evidence holds. Sacculina, the barnacle in the crab. What's proven? The anatomy, beyond any argument. The root system is real and mapped. The rootlets aimed at the nerve centers are real. The goblet organs pressed against degenerating nerve tissue are real, photographed, sectioned, described. The castration, the halted molting, the reshaped male abdomen, the brooding and climbing and fanning, all real, all replicated across species and coastlines. What's not proven? A single identified molecule of command. The feminization may be the demolition of a hormone gland followed by the host's own defaults. The parenting may be a stock program triggered by placement. The verdict the evidence supports. A parasite that rebuilds the host's hardware and then exploits software the host already owned. Renovation and trespass? Proven. Authorship of behavior? Still open. The killifish and its brain flukes. What's proven? And this is the strongest fitness result in the field. Experimental infection causes the conspicuous behavior, and the behavior delivers fish to birds at 10 to 30 times the background rate, measured in the wild. What's not? That the mechanism is targeted. A parasite that never touches the brain produces the same swagger, so the fluke's trick may be nothing more exotic than making a fish unwell in a way that shows. The verdict? Transmission enhancement, demonstrated about as well as it ever has been anywhere. Puppetry, unnecessary to explain a single data point. The amphipods. What's proven? Cysts in the brain flip the animal toward light and surface. Cysts elsewhere don't. The serotonin architecture is measurably rewired where the cysts sit, and injected serotonin alone reproduces the behavior in a healthy animal. What's not? Delivery. No one has caught the parasite dosing anything. The rewiring could be the brain's own inflamed response, concentrated where the foreign objects are. The verdict: the right neighborhood, the right transmitter, the right behavior, and a missing hand on the syringe. The tapeworm in the copepods and sticklebacks. What's proven? Timing that tracks the parasite's interests in both directions, caution enforced before infectivity, exposure after, and, in the two worm experiments, the resolution of a conflict between passengers, the infective worm's agenda overriding the others. Also proven, an energy theft so large that boldness and sterility both follow from the arithmetic alone. What's not? Any molecule, any pathway, any mechanism beyond the theft. The verdict: the strongest behavioral evidence for genuine, timed, interest-serving control anywhere in the sea, running on machinery nobody has found, and possibly running mostly on the budget itself. The cockles and the cataract flukes. Everything proven, because there was never anything hidden to find. Muscle replaced by cysts, lenses fogged by larvae, escape and vision subtracted, predation up, birds served. The verdict: manipulation by demolition. No debate, and therefore in a sense that should bother you a little. The purest form in this file. The parasite doesn't influence what the host wants to do. It edits what the host is able to do, and the want dies of irrelevance. The tongue, isopod. Proven. Entry, sex change. The tongue's slow death by strangled blood supply. The parasite's permanent installation as a working prosthesis. The fish feeding by pressing prey against a crustacean. Nothing about behavior, because nothing about behavior changed. The verdict: membership in a body, a foreign animal accepted by the host's own reflexes as a body part, because body parts are defined by position and function, and the isopod supplies both. The castrators, from the snail factories to the sterile king crabs. Proven. Reproduction deleted at population scale, hosts maintained as working assets, colonial parasites defending their territory with soldier castes, parasite biomass rivaling and beating the visible ruling class of whole estuaries, the energy of ecosystems rerouted through occupied bodies. The verdict: no minds touched at all, and the largest total transfer of biological control in this entire subject. If you rank these parasites by philosophical noise, the brain cases win. Rank them by what actually gets taken, and nothing comes near the ones that simply confiscate the future. Line the verdicts up, and the pattern is impossible to miss. Not one case, not a single one, shows a parasite writing new behavior into a host. That thing, the puppeteer implanting alien choreography into a hijacked mind, does not appear anywhere in the record. What appears instead, everywhere, is a short list of purely mechanical moves, used alone or stacked. Press an existing trigger, an egg-shaped sac at the brooding address, a body at the tongue's position. Cut an existing wire, a hormone gland dissolved, a foot eaten, a lens fogged. Lean on an existing dial, a serotonin system pushed off its setting, a stress axis retuned. Rob the budget and let the host's own intact decision-making conclude, correctly, that desperate measures are now rational. Or skip the animal entirely and become a piece of it. Triggers, wires, dials, budgets, parts, 5 moves. The whole haunted repertoire of the ocean's body snatchers, and every one of them operates on machinery the host was already running, because that machinery never had an owner, only inputs. So, Why do some ocean animals let another species run their bodies? The question can be answered exactly now in both of its parts. Why do they let it happen? They don't. Letting requires a gatekeeper. And the deepest finding in a century of this science is that there is no gate and no keeper. No checkpoint in a crab or a fish or a snail where the self stands guard and could, in principle, refuse. Permission never entered into the transaction. These bodies were simply accessible. And what does it mean to run their bodies? It means supplying inputs to open machinery. There is no command center to seize, which drops possession and puppetry and the entire language of minds out of the description. It means knowing. In the wordless way a lineage knows which lever sits where, what shape the socket takes, which molecule the receptor was always going to accept from anyone who offered it. The headline version of this story was that some creatures are so sophisticated they can enslave another animal's will. The evidence says something stranger, and it says it about every body in the sea, including the ones reading and narrating this. There were only ever mechanisms standing between a body and whoever learns to work it, magnificent, ancient, and unlocked, coherent for no deeper reason than that. Most of the time, nothing else had learned to reach them. A barnacle with no brain runs a crab. A worm with no eyes schedules a fish. And they do it because mastery was never required of them. And that is the real discovery hiding under 2 hours of parasites. Control of a living body is a keyboard that was never locked. And evolution, given time enough, teaches almost anything to type.